immunological agents. So before going into this topic, we need to know about what is immunity and what are the types of immune system, as well as how the immune responses are mediated. So what is immunity? So immunity, it is nothing but the resistance which is developed by our body against disease causing pathogens. Okay, again I am repeating, immunity is nothing but the resistance which is developed by our body against the disease causing pathogens. So there are two types of immune systems, like adaptive immune system as well as innate immunity. Means there are two types of immunities, like innate immunity as well as adaptive immunity. We know about the innate immunity. So the in the innate immunity, the immune responses will be mediated very quickly, whereas in adaptive immunity, the immune responses will take time. Coming into these immunological agents, see ma'am, in innate immunity, the immunity will be mediated by our own cells like natural killer cells. We know the process of phagocytosis, so that also comes under this innate immune responses. So coming to the adaptive immune responses, so those are mediated by the T cells and B cells. So coming to the adaptive immune responses, there are two types, like humoral mediated immunity as well as cell mediated immunity. So before going into the classification of this topic, we need to know about how the responses, means how the immune responses will be mediated by T cells and B cells. For example, humoral immune responses are there. So these humoral immune responses will be mediated by the B cells, whereas cell mediated immune responses will be mediated by T cells. So coming to the humoral immune responses, see how the B cells will produce humoral mediated immune responses means, see, whenever the antigen is presented or is entered into our body, so the antigen will be processed by the antigen presenting cell or any macrophage. So whenever the antigen enters into our body, the antigen will be processed by the antigen presenting cell or any macrophage. Then this antigen will be presented to the CD4 helper T cells. So whenever any antigen enters into our body, so the antigen will be processed by the antigen presenting cell or macrophage. So from there, the antigen will be presented to the CD4 helper T cells. So these CD4 helper T cells will be activated in the presence of IL-1, that is interleukin 1. So these CD4 helper T cells will activate the cytokines. Again I am saying, whenever any antigen enters into our body, so that antigen will be processed by the antigen presenting cell or any macrophage. After that, the antigen will be presented to the CD4 helper T cells, and these CD4 helper T cells will be activated in the presence of IL-1, that is interleukin 1. After that, after the activation of these CD4 helper T cells, so they will activate the cytokines. So these cytokines will undergo the proliferation and differentiation of B cells. So these B cells will identify the antigen, and this antibody will bind with the antigen and undergo the lysis. Like that, humoral immune responses will be produced. So coming to the cell-mediated immune responses, so these are mediated by the T cells. So there will be a precursor cytotoxic T cell, and it gets activated in the presence of IL-2. And these activated cytotoxic T cells or T lymphocytes will be matured, and then these cytotoxic T lymphocytes will identify the antigen and undergo the lysis process. Again I am saying, so in humoral immune responses, first if any antigen enters into our body, so the antigen will be processed by antigen presenting cell. So then the antigen will be presented to the CD4 helper T cell. Then the CD4 helper T cell will activate the cytokines. So these cytokines will be activated in the presence of interleukin 2. So interleukins are important for stimulation or for the activation of the immune responses. So these cytokines in turn undergo proliferation and differentiation of B cells. So these B cells undergo proliferation and differentiation, and they identify the antigen and undergo the lysis. So this is about the humoral immune responses. So next is cell-mediated immune responses. So there will be precursor cytotoxic T lymphocytes. So these precursor cytotoxic T lymphocytes will be activated in the presence of interleukin 2. After activation of cytotoxic T lymphocytes, they will identify the antigen and undergo the lysis. So next, coming to the classification of immunosuppressants. So there are various classes of immunosuppressant agents like calcineurin inhibitors. In that calcineurin inhibitors, the drugs include the drugs involved are cyclosporin and tacrolimus. And before going to this classification, we need to know about how the helper T cells will mediate or will initiate the immune response. See now, this is the cyclophilin, which is the intracellular protein. So this cyclophilin will activate a phosphatase that is known as calcineurin. What is phosphatase? That undergo the dephosphorylation process. Do remember the difference? Phosphatases undergo the dephosphorylation, whereas kinases undergo the phosphorylation. That means this calcineurin is a phosphatase. So this cyclophilin is the intercellular protein. So this will activate the calcineurin in the presence of calcium. After that, these phosphatase will be activated. So what is the function of phosphatase? It undergoes dephosphorylation, right? So this calcineurin will dephosphorylate the NFAT. NFAT means nuclear factor of activated T cells. So this nuclear factor of activated T cells will be translocated into the nucleus and starts the production of cytokine genes, that is IL-2 production. Again I am saying this cyclophilin is the intracellular protein, and this in the presence of cyclophilin, means in the presence of the calcium, it forms the calcineurin or it activates the calcineurin. So calcineurin is the phosphatase. So this calcineurin will undergo the dephosphorylation process, and this calcineurin dephosphorylates the factor known as nuclear factor of activated T cells. So this NFAT will undergo translocation into the nucleus and starts the production of IL-2 genes. So after production of these IL-2 genes, so this IL-2 will be diffused out of the helper T cell and binds with the IL-2 receptors and undergoes the T cell proliferation. And do remember that before undergoing the T cell proliferation, it will be done in the presence of mTOR. mTOR means mammalian target of rapamycin. So it is the key factor involved in the proliferation of T cells. So like that, immune responses will be mediated. So our drugs, like first class of drugs like calcineurin inhibitors, like cyclosporin. See, this cyclosporin will bind with the cyclophilin, which is the intracellular protein, and inhibit the activation of calcineurin. So if the calcineurin is inhibited, there will be no further steps takes place, and there will be no proliferation of T cells and no immune responses will be produced. And what is the main use of these immunosuppressants is they are important in the organ transplantation as well as in various autoimmune disorders. Why in organ transplantation means if any organ is transplanted into the other body, so that transplanted body won't accept the new organ or new tissue or any kind of cells. So it will produce the immune responses against that graft. Graft is nothing but the cell or organ or any tissue which is transplanted into the other body. So in order to suppress that immune responses, so these immunosuppressants are used. So this cyclosporin, it is the calcineurin inhibitor, so it will bind with the intracellular protein known as cyclophilin and inhibit the activation of calcineurin. And next drug is tacrolimus. So tacrolimus will bind with the another protein like FK binding protein. See, for the activation of calcineurin, we also require another protein known as FK binding protein. So this tacrolimus will bind with this FK binding protein and will inhibit the calcineurin activation. So if calcineurin or the phosphatase, if it is not activated, there will be no dephosphorylation process and there will be no production of cytokine genes. So if cytokine genes are not produced, then the other immune responses won't take place. So this is the other drug mechanism of action. So next class includes, we have seen about the cyclosporin and tacrolimus, where the cyclosporin will bind with the calcineurin, will inhibit the activation of calcineurin by binding with the cyclophilin, whereas tacrolimus will bind with the FK binding protein and inhibit the activation of calcineurin. So these are the calcineurin inhibitors. Next one is mTOR inhibitors. As I discussed before, this mTOR is nothing but the mammalian target of rapamycin. So this is the key factor which is involved in the proliferation of T cells. So drugs which comes under this class like sirolimus, everolimus, or 10 sirolimus will inhibit this mTOR. That is the key factor for the proliferation of the T cells. So if the proliferation of the T cells does not occur, then the further immune responses won't take place. So next class of drugs is anti-proliferative drugs. In the name itself is that they will inhibit the proliferation process. So if the proliferation is inhibited, there will be no production of T cells or B cells. So like that, they will suppress the immune responses. And glucocorticoids will inhibit the expression of MHC.